Hello everybody. Today's video is going to be a review on gene regulation in eukaryotes. Eukaryotic gene regulation is much more complex than prokaryotic gene regulation, and it isn't 100% understood. So what is gene regulation? Gene regulation allows specific genes to be expressed or not to be expressed. Basically, it is the reason why we have stomach acid in our stomach but not in our eyes. Although all of our DNA is pretty much the same, the genes on the DNA are expressed at different times at different locations. How does gene regulation occur? In both eukaryotes and prokaryotes, gene regulation is done by proteins. When does gene regulation occur? Genes can be regulated at many different levels along the central dogma. So we have pre-transcriptional, we have post-transcriptional, we have post-translational, but the most common one is during transcription itself. So that's the one that we're going to focus on today. There are different regions of DNA that affect gene transcription. We have enhancers, silencers, and insulators. Enhancers are regions of the DNA that interact with regulatory proteins to stimulate transcription of nearby genes. It can be located upstream, downstream, or even within the gene being regulated. Silencers are control regions of DNA. When transcription factors bind to a silencer, gene expression is repressed. They may be located thousands of base pairs away from the gene that they control. Insulators are stretches of DNA that blocks the interaction between enhancers or silencers and the promoters. They prevent a gene from being influenced by the activation or repression of its neighbors. They can be as few as 42 base pairs away. So we have repressors and co-repressors. Repressors are used to suppress transcription by binding to silencers and recruiting co-repressors. Repressors recruit co-repressors with two different functions. Either the direct way, where co-repressors can block RNA polymerase, or the indirect way, where co-repressors can tightly pack those histones so that it cannot enter and that genes cannot be regulated. Indirect repressor proteins can interfere with the function of an activator in four ways. Competition, which is due to overlapping binding sites, quenching, which is when the repressor binds to activation domain, cytoplasmic sequestration, which is when it binds to an activator and keeps it in the cytoplasm, or heterodimerization, which is when it binds to an activator and prevents homodimerization. There are three different ways of transcriptional gene regulation. We have coordinated, RNA stability, and non-coding RNAs. The coordinated is when a single stimuli may turn on multiple genes, or a single gene may be regulated by more than one response element. So either one transcription factor binds to multiple genes, or multiple transcription factors bind to one gene. Then we have the RNA stability. This includes the removal of the cap. The enzymes eat DNA from 5' to 3', or from 3' to 5', or the nonstop decay, which is independent of removal of cap or tail, and so it chews from the middle. Lastly, we have non-coding RNAs. This involves the small non-coding RNA or the microRNA. Although eukaryotic gene regulation is very complex, it and prokaryotic gene regulation share some features. An example is in both types of cells, DNA binding proteins influence the ability of RNA polymerase to initiate transcription. Here's some of the major differences in gene regulation in eukaryotes and prokaryotes. Prokaryotes are organized into operons and are transcribed into a single RNA molecule, where eukaryotes have their own promoters for each gene and are transcribed separately. In eukaryotes, DNA must unwind from the histone proteins before transcription can take place, and the presence of the nuclear membrane in eukaryotes separates transcription and translation in time and space. For the sake of time, that is it for today. Thanks for watching.